There we go. All right, welcome everybody to day one still of our bioinformatics workshop. This is the part now where we're going to get into talking about amplicon sequencing and have a hands-on tutorial led by Kristin Yoshimura, who's currently finishing up her PhD at the University of Delaware. Yay!
Thanks. Hey guys. So I get the pleasure of talking to you about amplicon sequencing today, mostly because Ben didn't want to. So for now, I'm just going to make this big because I'm not doing anything. Okay, cool. So just to orient you guys to what we're going to be doing, we're going to start off with the hands-off portion where you guys can just sit back, listen, take it all in, ask questions if you have them. I'll try my best to answer them. I'm going to talk about a brief background on amplicon sequencing. What is it? What information can we get from it? Go into talking about experimental protocol design. So what things do you need to consider when doing amplicon sequencing? I'm going to talk about analysis methods, what we've done in the past, how the field is kind of evolving and changing, and what the best practice methods are now. And one of those methods is QIIME2. So I'm going to give you a brief introduction to QIIME2 before we go into the hands-on portion where we're going to be doing a small tutorial of an example QIIME2 workflow. So what are amplicons? Amplicons are the targeted amplification of a specific gene. And that can be any gene that there's primers available for or that you can develop primers for. And these can be functional genes. So say you wanted to study nitrogen fixation in sediments, you might choose to look at the NIF-H gene specifically, or if you wanted to study methanogenesis, you might specifically want to isolate and amplify the MCRA gene, and then look at the diversity of that gene to tell you something about those processes in the environment or the players in those processes. So you can look at functional genes, but you can also look at phylogenetic marker genes. So for bacteria and archaea, we would look at 16S rRNA genes. Eukaryotes, we'd look at the 18S rRNA gene. And people who study fungi a lot of times prefer to study the ITS region, the intergenic transcribed spacer region, between the small subunit and large subunit rRNA genes. So over here, we've got this 2D structure of the 16S rRNA. I'm going to be talking about 16S because I study bacteria and archaea, but a lot of these considerations that I'll be talking about, you can also apply to 18S or other functional genes. And so the 16S gene has conserved regions and it's got hypervariable regions. So the conserved regions are pretty much the same for almost all bacteria and archaea. They're really important in structure and function. And then there's these hypervariable regions, and that's where we get a lot of the phylogenetic information from. So if you took the gene and you stretched it out end to end, you'd kind of get something that looks like this, where we'd have these conserved regions in blue and the hypervariable regions in red. There's 9 of them. And the gold standard in the field would be to sequence that entire 16S rRNA gene, roughly 1,500 base pairs long, and you do that by cloning it and Sanger sequencing it. But there's some cloning biases and primer biases I'm going to be talking about later, but it's also a very low throughput method. So historically, papers that were using Sanger sequencing, they might publish on 20 clones, 100 clones, but they're trying to tell an ecological story from very few sequences. And now we can get around that with these NGS methods that are much more high throughput, but a lot of these newer methods, you can only get smaller sequences from. And so we're only able to sequence smaller pieces. We can't do the whole 1500 base pair region. And so we kind of focus on some of these hypervariable regions that might be able to give us some phylogenetic information. So depending, well, you might want to choose the hypervariable region that you're targeting based upon the organisms you're looking for. So different hypervariable regions can lead to varying results. So this was a study done where they looked at a couple different methods for 16S sequencing. They looked at the V3 region, the V6 region, and then they did a full length. Something to note is that if you look here at the end numbers, the full-length sequencing, the Sanger sequencing was a lot lower throughput. It's only 5,500 sequences that they had, where we're looking at 300,000 for these, for the tag sequencing. And overall, it tells a general story that's pretty much the same from method to method, but you can tell there's some small differences. So there's no region that's going to capture the phylogenetic differences for every single organism. So to get into some considerations for protocol design, so for those of you who aren't familiar with the Earth Microbiome Project, it's this big effort to try and look at the entire diversity of microbes on our planet. It's impossible for one researcher to go out and do that. And so they kind of crowdsource it. So if you want to be part of the Earth Microbiome Project, you can go out, do your study, sample it, and then submit your data to the Earth Microbiome Project. But in order for your sequences to be able to be compared across studies, there has to be a method that every single person uses. And so they've outlined their recommendations for how to do amplicon sequencing. So they look at what data you need, what metadata you need to be able to accurately describe your data. They go into different extraction protocols, what primers to use, and then how to sequence your how to sequence your data. But outside of that, there might be additional considerations you want to think about depending upon the type of sample you have or the organisms that you're looking at specifically. And so I'm going to go into that a little bit. But before that, I want to talk about sampling a little bit. So when you have your project, you're going out and you're trying to figure out how many samples you need to sequence. Something you want to take into consideration is having enough samples for biological and technical replicates. And the idea behind that is you want to make sure that you're getting a representation of the environment that you're sampling in, or you can environment or human associated or other organism associated. And also you want to make sure your results are replicable. Was there an error somewhere in your protocol and you're getting variation because something went wrong while you were doing your sequencing or something? So the goal behind that would be to have good statistical support of your results at the end of it. And it would be good practice to use positive and negative controls. An example of using a positive control would be if you're designing your own primer set and you want to make sure that it's hitting certain organisms that you think it should hit, you might want to use something like a mock community and so you can actually say, yes, it hit all of these organisms that I'm looking for. And then negative controls, we know that there's contaminants in extraction kits, there's sequencing contaminants, and so using negative controls can help you sort of pinpoint where your contaminants might be coming from. So you're not saying you have a bunch of Phi-X in your sample. No, that's probably from the sequencing center. Okay, so DNA extractions. There's tons of different methods out there. There's a bunch of kits available. You can get water kits, soil kits, tissue kits, and they're all great for different types of different types of material that you're extracting from, but you can also extract by creating your own all your own reagents, doing like freeze thaw methods or C-TEBI method. But all these extraction protocols have slightly different efficiencies, and your efficiencies will also depend on what kind of organisms you're trying to extract from. So some things that you might want to take into consideration when choosing your extraction protocols are what type of sample are you extracting from? So if you're extracting from an environment that has a lot of metals in it, a lot of organics, other contaminants, it might interfere with the extraction process. So you might have to alter that extraction method to deal with that. And also, what microbes are in your sample that you're looking for? Is it gram positive or gram negative? So is there something in the cell membrane that's going to be harder to extract the DNA from? If you're trying to look at archaea specifically, you might want to add a proteinase K step because we know that they have different cell membranes. If your organisms might be sporulated, that might also affect your extraction efficiencies. So you want to take all of these into account to make sure that you're going to be extracting the best DNA that you can. So after you've extracted your DNA and then you're going to go and sequence it, you have to figure out what primers you're going to use in that sequencing method. All primers have primer bias, so there's no primers out there that are going to amplify every single thing that you're looking for. But my advice is just know what that primer is, primer bias is. So these primers don't pick up on these certain organisms or have a bias against them. So also when choosing your primers, you might want to consider what amplicon size you need. So that might be kind of limited by your sequencing method. How long of a fragment are you able to get versus how long of a fragment do you need for that particular gene or that particular hypervariable region? And one way that scientists can kind of overcome small fragments is try and do paired end reads. So you have a forward read and a reverse read. You trim off the bad quality bases, but then you still have enough to overlap and you can get a longer fragment that way. And also note that there's variations in amplicon size based on the organisms present. So just to show you an example of that, here Sinclair et al. Looked at the V3, V4 hypervariable regions and noted differences in the size of their amplicons. So they had a smaller size here that was produced by archaea, and then there were chloroflexi produced this mid peak, and then Bacteroidetes and some others produced this larger peak, and things like the proteobacteria had a distribution across these different sizes. And so here we can see maybe a 50 base pair difference in size for the V3, V4 region. So anticipate that when figuring out how long your amplicons are going to be. And just to drive this home, that there's primer biases. Here, Parada et al. Looked at two different primer sets, this 515FY806R and 515FY926R. And they looked at the percent abundance of these different, or the relative abundance of these different community members. And you can see that there's some drastic differences in some of these, for some of these microbes. And okay. So to go into sequencing platforms a little bit, we've been talking a lot about Illumina sequencing, MiSeq, HiSeq. That's very popular these days. Ion Torrent, some people still use it, but not as much. And Lee Kirchhoff just came out with a paper where he sequenced the entire 16S RNA gene using Oxford Nanopore. So that might be a new method that start to emerge. But the sequencing platform that you want to choose might depend on the size of your amplicon. So Illumina HiSeq will give you maybe 150 to 250 base pairs, where MiSeq can give you up to 300 base pair reads. Depth of sequence coverage, and I'm going to go into that a little bit more in a second, but if you choose Illumina MiSeq, you might be getting tens of thousands to hundreds of thousands of reads, whereas you're doing HiSeq, you're getting hundreds of thousands to millions of reads. So how many sequences do you need to get? And then the number of samples you have, how many samples can you sequence at one time? So we have this method called multiplexing, where we're talking about this. Every sample will get its own barcode attached to it, and then you can pool your sample and sequence it all at the same time. So you're getting more bang for your buck. And then later we demultiplex it. So we separate the reads out into their different samples later on in the process. But how many samples you're allowed to multiplex together might also be a consideration for choosing a certain sequencing platform. So I just mentioned sequencing depth. How many sequences do you need? That's a question I've asked a lot. So how many sequences am I going to ask the sequencing center to give me back? And that highly depends on what your project is and what your question is. So what type of sampling environment do you have? Is it complex or is it simple? So if you're in an area like acid mine drainage, it might be a more simple community, whereas soils and sediments have a much more complex community. So you might need a deeper sequencing depth to look at more complex environments. And are you looking for dominant or rare organisms? So this idea of the rare biosphere, there's a couple organisms in a community that are really abundant, but then there's this long tail of many, many organisms that are really rare. So are you just looking at the main community members who's really contributing the most to your environment, or are you looking for something specific and rare and you're really trying to find those things that are only occurring a couple times in your sample? So you might have to do deeper sequencing in order to get at those rare members. Say for most projects, about 10,000 sequences per sample is sufficient. Again, it depends on your project, but that should be a ballpark estimate of things to shoot for. But then you can use things like rarefaction curves or the Chow-1 richness estimator to estimate the capture the total diversity. So if you're using rarefaction curves, which I'll show you what those are in a second if you're not familiar with them, do they start to level off, or are you still in that exponential phase? So this is what I was talking about when I mentioned the rarefaction curve. So you subsample, and every time you subsample, you count how many different organisms you're finding. And so as you increase the number of reads, you're increasing the number of different organisms you're seeing, but eventually these curves will start to level off. And that just means that you're not seeing new organisms the deeper you sequence. So you can kind of say with some confidence that you captured most of the diversity. And the London et al. Paper that I referenced on the previous slide looked at how many sequences you need to be able to accurately estimate alpha and beta diversity. So alpha diversity is within sample diversity. It's basically all the metrics used to characterize a single sample. So things like number of organisms, coverage, different diversity metrics, Shannon, Simpson, face phylogenetic diversity, and microbial richness. So they did some tests and they figured out that you need about 5,000 sequences in order to explain 90% of most of these alpha diversity metrics and about 80% of the trends for Chow-1 and richness. And then when looking at beta diversity, you needed only 1,000 sequences to estimate the trends or to explain the trends for 90% of the beta diversity. And beta diversity is between sample diversity. So how closely related is this one sample to another sample that you're studying? And you can look at that through like principal coordinate analysis plots, dendrograms, where you're trying to figure out, okay, so these, I know it's really tiny, but these sediment samples here are all more closely related than all of these nodule samples here. So getting into analysis methods, previously we had done the OTU method. So OTU stands for Operational Taxonomic Unit. And basically what that was is we said that anything that's 97% similar to each other, well, it's operationally defined. You as the researcher can define what that percent cutoff is. It's commonly 97%, but you can also change it 99, 95, 90, whatever you want. But whatever that OTU threshold is, you say anything that's like 97% similar to each other here is we're gonna call that one thing. And here, these are all 97% similar to each other. So we're gonna call those one thing. The reason why we did that is there's inherent stochastic error in these PCR steps and sequencing. So when you add the primers on and you add the barcodes on, you have you introduce some error there, and then there's a little bit of error in the sequencing itself. And so in order to mask all of that error, that's why we look at it at a 97% level. So in that 3%, we're masking some of that error that was introduced. As a side note, sequences that are 100% identical to each other can have different genomes. So there could be variation somewhere else in the genome that doesn't occur in the 16S rRNA gene. So keep that in mind too. So there were a couple different choices of programs for OTU picking methods. QIIME and Mother were very popular. There's other methods as well, like uparse. And they had several different OTU picking strategies. I won't get into it too much because we don't really do this anymore, but there was open reference, closed reference, and de novo. It's just different methods on if you're picking your OTUs based on a database or just based on your data set itself. But this OTU picking strategy, or these OTU picking methods, it's hard to compare study to study because, especially if you're looking at de novo OTUs, those seeds that they picked can change based on your study. So if you choose a seed and say anything that's 97% similar to each other as an OTU in one study, that seed might be in a different place in another study. So it's hard to compare OTUs across samples. There was also another method called oligotyping, and it was looking for diversity at a sub-OTU level, so a finer detail than the 97% cutoff that we're looking at with OTUs. And this method was really useful for discerning differences in sequences that were very similar to each other. So looking at different strains, or certain strains more abundant at different times of the year, and so forth. But we're also moving a little bit away from the oligotyping too. Yeah.
How does the oligotyping differ from just saying your OTU cutoff and higher the Chinese? Or you can like keep increasing how similar they have to be.
Right. So the oligotyping method was looking at very subtle variations among the sequences. So it wasn't like a percent cutoff. It was looking at the nucleotides in cells, and the position of the nucleotides was really important as well. So it's just a different, But we're moving away from both of those methods and getting into this ASV method. So ASV stands for Amplicon Sequence Variant. And so, as I was mentioning with the OTU picking methods, the way that they dealt with error was to mask it by grouping everything at a 97% identity level. So this ASV method works to infer the sequence before the introduction of errors in the amplification and sequencing processes. So its algorithm basically tries to back calculate and figure out what that original sequence was without error. And so when these methods call an ASV and they have single nucleotide variations between different sequences, those are actual differences. So they're two different sequences and not, so it's hopefully correcting for any errors. So there's a couple programs currently in order to look at ASVs. And Data2, DeBlur, UNoise is another one, or UNoise 3. And Nearing et al. Compared these three methods to try and figure out, was one of them better than the other? This is in PeerJ. It hasn't been peer reviewed yet, but I thought it had some good points to make. So Data2 appeared to be the most sensitive to rare ASVs. So going back to what is your question? Are you looking for dominant things or rare things? If you're looking for those rare organisms, you might want to consider using Data2. But Data2 is also the most time and memory intensive denoising programs of the three. For the data that we're going to be working through, it took 4 days for Data2 to run.
What do you mean by a denoiser?
So these are the programs that are going to pick the ASVs. DeBlur, so if you're limited by the amount of memory you can use, DeBlur might be a good option. So DeBlur always uses the same amount of memory, but the amount of data that you have as it increases, the time it takes to run will increase. But that time is still less than it would take Data2 to run. And one thing to note is Data2 and DeBlur are both open source programs and implemented in QIIME2, whereas UNoise has 2 different versions. There's a 32-bit version, which is open source, but their 64-bit version is behind a paywall.
Has a comparison of richness been done comparing OTUs versus ASVs? And do they come up with the same trends or no?
So what I've heard is that the OTU method kind of overestimates how many OTUs there are. And so these programs will give you back fewer ASVs than OTUs.
But that's at a single nucleotide variation, and yet you'll still come up with fewer ASVs than you would OTU when it's at a coarser resolution?
That's what I've heard from people who have looked at it both ways. Yeah. So getting into QIIME2. So again, we can run both Data2 and DeBlur in QIIME2. It's a Python program, and the only other thing you'd need to run it would be Anaconda or Miniconda. Those are the only requirements that you need to run it. And if anybody here has run QIIME before, it's much different. QIIME2 is very different from QIIME1. And the first difference is that there's multiple interfaces. So there's this graphical user interface, Q2 Studio. And if you have friends who need to run microbiome analyses, but they have no command line, and they come to you and they say, hey, can you help me run this? Instead of taking like 3 days to teach them Unix and how to run command line before you even get into doing any analyses, you might want to suggest using this GUI interface for them. It's for scientists who need to analyze microbiome data but don't have any experience in command line. So there's boxes that you click and drop down menus, things like that. But it can only be run on like your laptop, a local computer. So you can't do it through a terminal window. The next is command line interface. So that's what we'll be doing. That's what all of you guys would be using. It's for power users, including those with command line experience and have access to clusters. Again, you can't use that GUI interface if you're working on a server. And then it also has an application programmer interface, Artifact API, but that one's more for plugin developers, data scientists, and for people who are developing other uses for QIIME2. Another big difference between the original QIIME program and QIIME2 is that all of the data is stored as artifacts. So these QIIME compatible artifacts would be the .qza and .qzv files. .qza stands for QIIME zipped artifact. .qzv stands for QIIME zipped visualization. These files contain your data. So if it's an FNA file or a biome file, it would be stored in that QZA format. It would have a unique file identifier, but this really cool thing is it has provenance. So when you open up this file, you can see all the steps that have been done to get to that file. So if you forget, you can just look at that provenance and figure that out. But that also requires import and export of data into and out of QIIME compatible formats. So you can't just take your sequence data and start running data2. You have to import it first, then run it. And then if you wanted to use the data outside of QIIME2, you'd have to export it again. So QIIME2 is still being developed. They come out with a new version about every 2 months. It's very annoying every 2 months having to uninstall and reinstall new program, but it also means that they're still developing it and making it better. So there's certain things where I'd go on the QIIME2 forum and say, hey, I need to do this. Is there a way? And they would come back to me, not yet, but thanks for letting us know and we'll work on it. And maybe in a future update, we'll have it for you. So it's still being developed, but let's say you have downloaded it or installed it, but you forget which version you have because there's a bunch out there. You can just type into your terminal conda list and it will tell you which version you have. And then you need to know what version you have in order to activate the program. So to activate, you'd type out source, activate, and then the version of QIIME2 that you have. And then you can test the installation and make sure you've accurately installed it and you've activated it by typing QIIME -- help. So originally QIIME had these Python commands that you would run. QIIME2 works more based on plugins. So all of the functionality in QIIME is through a different plugin. There's one for data2, there's one for deblur. Every different step has a different plugin that you'd use to work through it. And developers can also create third-party plugins for QIIME2 as needed. So somebody needs a new function, somebody else can create it and then make it available to the public. So to look, and you can go to this website here, or this address here, to look at all the available plugins currently. So to run commands in QIIME2, you would call QIIME, so you'd call the program, and then you'd call the name of the plugin that you're using, and then any flags that are required. So for example, you'd run QIIME data2, and then let's say I didn't know what parameters, what flags I needed to run the program. I could write -- help and it would print out a list of all of the optional and required parameters that I need to use with that plugin. So let's say you go home and you forget everything that I've taught you, and but you still want to use QIIME2, you can go through some of these tutorials were really helpful for me when I was first starting and figuring out how to do this. Likely, there's no one tutorial on the website that's going to fit your needs for your specific data perfectly. You're going to have to mix and match, but there's pretty good documentation, as Ben was saying before. Some of programs have everything is documented for you. Some things there's very little. I'd say this one's fairly good. And then there's some things that, you know, Google, Stack Overflow, the QIIME2 forum are additionally very helpful to me. So if you guys want to get a snack before we go into the more hands-on portion, feel free. Ben says that you guys have to eat the snack because he paid for it. So before we move on, does anybody have any questions that I can try to answer about 16S or protocol or QIIME2 so far? Crickets. Great. Okay. Also gonna make this smaller so we can see the terminal.
You can make it a little bigger if you want, because you don't switch back and forth between. You don't see. There's a click on the command line. Go and hide all of that stuff. Yes,
Just like that.
perfect.
All right, we all good? Ready? All right. So getting into QIIME2. So these are some of the steps that that you would do when doing it. So, and I'll go through some of these more detail as we go along, but just briefly. So we need to create a metadata file. So This is a file that contains your sample ID names. It might be similar to a mapping file if any of you have used that before with your barcodes and primers on it, but add any other additional columns like body site that your samples were taken, or time of year, or any other metadata you want associated with their samples. And then we have to import that data into a QZA format. And then depending upon what type of import you do, what format your data is in, you may have to demultiplex the data. So you take that jumbled mess that comes out of the sequencer and we sort it based on the barcodes that we use. And then I didn't put it here, I forgot, but if your sequences still have the barcodes on them, sometimes the sequencing center will take them off for you, sometimes they won't. So you might have to use a program. So CutAdapt is Ben just talked to you about. That is implemented in QIIME2, so you can use that to take the barcodes or other adapters off of your sequences. Then you'd go into denoising and QC filtering, so using data2 or deblur to pick your ASVs and also quality filter your data. You'd have to build a phylogenetic tree then of your samples, and we use that phylogenetic tree for some of these diversity metrics that you can do in QIIME2. And then the next thing you would do would be to assign the taxonomy. So QIIME2 suggests that you train your classifier. So what that means is you have a database that you're using to assign taxonomy to your sequences, and to get a better identification, you take your database and you trim it to just the hypervariable region that you're looking at. And so you're not looking to try and assign data from the whole database. It's just the part that you're looking at. There's also a step to filter your data in different ways. I have taxa here, but you can do it by sample. Other ways we'll get into that. And then if you want to use any of this data in another program, say you don't like the bar charts that QIIME2 puts out, you want to use R instead, you'd export that data and then you can use it in another program. So is everybody ready? Your terminals are woken up? Okay, so the first thing we're going to do is navigate to the QIIME2 tutorial directory. So we're going to use the cd command, cd/home/c-debi/ecogeo/QIIME2 tutorial. And if you want, you can just copy that link and paste it in here. There we go. Next, we're going to activate the program. So we're going to type out source, activate, and then the version that is installed in the virtual machine is QIIME2-2018.4. Hit enter, cool. And then test whether or not it is properly activated by QIIME-help. And you should get a list of these different plugins. Is everybody there? Everybody's got it working? All right. So the first step was to create a metadata file. It's typically a TSV file. It can also be a .txt file. And this link down here, if you go to that, will give you some more guidelines on creating your metadata file. But if we look at the metadata file we'll be using here, it's a very bare bones one. It doesn't contain much data, but to give you an idea, if you do less and then metadata, you can start typing like MET and then tab. And see it's a TSV file, and we have a couple categories here. And then to get out of that, again, press Q. So I'm going to talk to you about importing data a little bit. This was probably one of the most confusing steps. So there's a couple different ways to import your data, and it's all based upon what your data looks like. So when your data comes back from the sequencer, it can be multiplexed, it can be demultiplexed. Some of it depends if you get paired end or single end reads. Sometimes you get back a mapping file, sometimes you get back a barcodes file. So there's different ways to deal with that depending upon what you get back from your sequencer. So the first method is the EMP protocol method, so the Earth Microbiome Protocol that I talked about earlier. And this is for data that is multiplexed. So it's that jumbled mess that comes out before it's sorted by barcodes. And it can be single end or paired end data. And then the other thing you need is a barcodes file. And so that is what this is. If you have a file and you open it up and it looks like this, that would be the associated barcodes file. So if you have those two things, multiplex data and barcodes file, you can import it using the EMP protocol method. But if your data is multiplexed, but say you get back a mapping file and not a barcodes file, you're going to need to do some work to that data before you can import it. So you're going to need to demultiplex the reads in a different program first before you can import it, and then import it using a different method. So for example, you could use the QIIME program, and you can use these Python scripts, split libraries, FASTQ. So that is the demultiplexing step, sorting it by barcodes into its different samples. And then split sequence file on sample IDs.py. And what this script will do is it'll take that demultiplex sample that you have and make a separate file for every sample you have. And that's going to be important for the manifest protocol. So the FASTQ manifest protocol is the other way to import your data. And this requires demultiplexed single or paired end data. And the files can have an extension of .fastq, .gz, or .fastq. But you have to, the demultiplex data also has to be in every sample, needs to be its own file. So that's important. And then you would create a manifest file that kind of follows this format here. You'd have a header, sample ID, comma, no spaces, absolute file path, comma, direction. So that's your header. And then underneath, you have a list of all of your samples. So it would be sample name, the absolute file path, not a relative file path, absolute file path to the sequence file, and then the direction of reads. So if you're only working with single end data, it would all be forwards. If you're working with paired end data, your R1s would be forward, R2s would be reverse. And so you'd list it out. So sample 1, R1 forward, sample 1, R2 reverse, sample 2, R1 forward, sample 2, R2 reverse. And so you'd list out every single sample you have, and then if it's paired end, forward and reverse. And so not only does your data have to be, every sample is its own sequence file, but the forward and reverse reads need to be in their own files as well. Which brings me to my next point. If your data is paired end and demultiplexed, but the forward and reverse reads are interleaved. So that means in your FASTQ file, you have read one forward, or sequence 1 forward, then sequence 1 reverse, and then sequence 2 forward, sequence 2 reverse. You'd have to split those into one file with forwards, one file with reverse. And then you could import it in this FASTQ protocol. Did I lose anybody, or does anybody have data that looks differently that they're working with? I know that was probably one of the most confusing steps for me to figure out is, okay, what does my data look like and what format do I have to get it into? So there might be some data manipulation on the front end before you can import.
Could you define demultiplex?
So it's when, so your data is kind of all jumbled up when it comes back. So so every sample gets its own barcode, you pull them together, you sequence it, and it comes back as a big mess. And then demultiplexing, it sorts it by barcode. So all of these are one sample, all of these are the other sample.
What exactly is if you don't have a barcodes file, you say you have a mapping file. What exactly is a mapping?
So a mapping file is kind of like a metadata file. So it would have, so I've gotten this back from certain sequencing centers. It would have your sample name and then what barcode was associated with each sample, what primers were used, things like that.
Because I've gotten some sequencing back where actually within like the line with the carrot, I actually have the barcodes there, but I actually did the barcoding myself. So.
It's just,
So I guess I made my own mapping file.
yeah, it's just one of the ways that I know that I've gotten data back. So I wanted to talk about it a little bit. Okay, is everybody on board so far? Okay, so this part took maybe 10 minutes and we didn't want to waste time with how long that took. So we've already imported the data for you that we're going to be working with, but just to go over what we did, so we type out QIIME, tools and import. So that would be the plugin we're using. Type sample data paired end sequences with quality. If you only had single end sequences with quality, you'd have to note that. So these sequences with quality means we're working with a FASTQ file, not a FASTA file. Input path, so I'm doing the manifest protocol. So I've created this manifest.csv file that, you know, I told it what my samples were and what the path was to my samples. Output path, demux. So just to note, if you guys go home and do this and it's a little bit different when I was doing it, I'd have to put the extension here, so demux.qza, and then we were testing it in this virtual machine and it was outputting demux.qza.qza, so it was adding the extension itself. So I've seen it happen both ways and I'm not sure why one or the other happens. So test it to see whether or not you have to add the extension on your output files. But in this virtual machine, if you see this output path, any of this code will not have the extension on it because QIIME2 is going to put that extension on. But that was just a little side note. And then source format, we talked about different quality scores that we can use. So this one is FRED33. So we're going to note that as paired end FASTQ manifest FRED33 in the source format. So to make all of this code a little easier to look at, I've added, so every line I've delimited with this backslash. And so that's the same thing as saying everything is on one line. So if you're typing this into your terminal, all of this data can be on one line and everything is just separated by a space. But if you copy this and paste it directly into your terminal, it will act the same way. So you can do it both ways. So I've imported the data, but let's look at the data. So we're going to do QIIME demux summarize. And then input the data --i -data. So we're inputting data. And that is going to be the demux file that we imported. And then we want to output a visualization file. So this is going to come out as a .qzv file. And then we can use QIIME tools view to then view what that file looks like.
You have to go in one.
Oh, it's GZA, I think. I think it's wrong. Yeah, okay. So it's wrong on the PowerPoint. This should be a G because it's zipped. Okay. And we'll wait for that to run.
What's a GZA file? I still got it.
Still got it there. Okay. It was zipped because it was a big file. So you'll get this confirmation, saved visualization to demux.qzv. And let's look at it. So QIIME tools view, and then view that QZA file or QZB file. It took a little while to create that file, yeah. So we'll wait till most people have got it.
Can you switch back to the command that allows you to open this up after the first one?
Trying. There we go.
So that was
Oh, other way.
the QIIME tools you
Yep, right here. So how are people doing? Have you guys been able to open this yet? No? Is it still running or are you getting an error? Okay.
You do that. It opens up to.
Oh, yeah.
No.
you need to have a $3 soda.
Yeah, that's very
How to get it?
Oh,
So-
they need this.
So that's what I typed in QIIME tools view demux dot. So what I'm doing is I'm using the tools plugin to view the QZV file that we created from the QZA file. So while it's running, I'll just go over what this file is supposed to look like. So we get some summary of our sequences. So we have between 135,000 and 274,000 reads. And then we can look at a histogram of that. So one sample had this many and 2 samples had this many, so forth. And then we can look at our 6 samples individually and see how many input sequences do we have. And the other thing then is you can go into this interactive quality plot. And now we're going to look at this in order to inform our QC steps. So when we were doing the Illumina quality sequencing, we were looking at these quality plots and trying to determine where we wanted to cut our sequences. And so we're doing a really similar thing here. So we have our forward reads here and our reverse reads here. Our reverse reads right off the bat look worse than the forward reads. But the forward reads look pretty good. There's a slight drop off in quality at the beginning. And then there's a big drop off in quality around here, maybe around 280 base pairs. And the reverse reads, the drop off in quality happens much sooner. But then you can also look at this nifty thing where, you can go along and look at specifically here, what are the stats here? We can look at that box plot, the lower whisker, middle of the box. What are those Q scores that are associated with this particular region? And so using that Q score, we can say, oh, that's a good Q score, I'm going to keep it. But over here, the Q scores drop significantly. So we're going to get rid of it here. So did everybody get it? So are most people on track now? Okay. Okay, and then you can just type Q to get back and can move forwards. So just something to note that this QIIME tools view command will not work in a headless environment. So if you're working on a server cluster, you can't use this command to visualize these files, but visualizing the files is still really important. So how do we get around that? You would transfer your file to your local computer, and then if you had QIIME2 installed on your local computer, you could then use the QIIME tools view command from your local terminal. Or, because I don't have QIIME2 installed on my computer, this is what I do, is this right here, https://view/QIIME2.org. If you type that in, you'll get a web page, and this will be on the top of this web page, and you can drag and drop your files into the web page, and then it'll open up that visualization too. So multiple ways to do this. So the next step would be to use data2 to do the quality control and denoising. So this was the step that I told you took 4 days. And so instead of sitting here for 4 days, I've done it for you, but just I'll talk about what we're doing. So we're calling QIIME, data2, and then denoise paired. If you had single end reads, you'd do denoise single. We're inputting demultiplex seek. So we're importing or inputting that demux.qza file. And then all of these are those parameters. So we decided where we're going to trim. And so, oh, sorry, this is where we're going to tell the program at what base pairs to trim. So trim left, forwards, trim left, reverse. So that's trimming at the forward end of the sequence on your forward and reverse reads. So I decided to trim off a few base pairs in the beginning because they weren't as good quality and I was a little suspicious of them. And then this P trunk length, so truncation length is what that stands for in your forward and reverse reads. And so the forward reads, I'm saying cut it off at 289 base pairs. They were pretty good quality, just that very few last base pairs weren't very good. But my reverse reads, the quality dropped off much sooner. So I'm gonna have my reverse reads be shorter. Yes?
Did you decide on that based on the visualization?
Yes.
So you just said, okay, well, this looks crappy. I'm going to just cut those.
Yeah, so I used that and then like put my mouse over those different places and I looked at what the Q scores were. And so all based on that, these were the decisions I made. You can make your own decisions based upon how stringent you want to be with your data. And then it's going to give you 3 outputs. It's going to give you a table. So a table of all your sequences, representative sequences. So it's going to pick all of those different ASVs and say like 300 sequences are in this ASV. It'll just pull out one. And then that'll help us in the taxonomy assignments. It'll make it much quicker. You only have to assign taxonomy to one sequence instead of all 300. And then it'll match later on. And then this denoising stats. So it'll tell you how many sequences you had at every step. And so you can see where, if the program gets rid of some of your data, you can see at what step all that data went. So again, this is the most time intensive step depending upon the size of your data. So the goal in the QC and denoising step would be to maximize the amount of output data with good quality. So sometimes if you want more data, you might be a little less stringent on the quality. If you're looking for really rare sequences or you want to determine the difference between very similar sequences, you want to make sure that it's really good quality before you move on. So you might be a little more stringent. And then if you're using paired end reads, you want to make sure that the trimmed reads overlap so you can get that larger contig. So things you want to think about, how long is the amplicon length of your primer set? So how long is that gene you're looking for? And is there a variation in that gene length? And what is that variation? So how long do the reads need to be? How long are the sequence fragments? And then once you cut that data, so once you trim off the bad parts, is there still enough overlap in order to make that longer read? So I told you we had 3 different outputs: the table, which we don't really need to look at, the representative sequences, but we are going to look at the denoising stats. So let's do that. Do QIIME, metadata tabulate. So we're just putting all this data into a table. We're going to input that QCA file. And you can start typing and then tab completion, make your life easier. And then we're going to create a visualization. Oh, I spelled that wrong. And call it denoising stats. And then when it's completed, you'll get confirmation that it saved it to this .qzv file. And then we can view that file again. So QIIME tools view, and then that QZV file. And so what we can see is that, so we input the data, and this is how many sequences we input. Then we filtered it. So we filtered out any of those sequence contaminants, so like phi-X reads. Then was the denoising step. So choosing all those ASVs. And so we didn't lose any data because it's just sorting them at that point. merging the paired end reads together, so we're losing some data. And then the program looked for chimeric sequences, so artificially produced sequences that aren't real. And so we lost a lot of reads there. So we started with 274,000 reads and we're winding up with 41,000. So lost a lot of data, but we still have enough and I'll show you why in a little bit. All right, Q to get out of that. Is everybody, yeah?
Example, what primers did you use and how many base pairs is available?
So, okay, let me think about this. So I used, they were V4 primers. The region is about 250-ish base pairs. And so if I had, my forward reads were like 280 base pairs and my reverse reads were like 200 base pairs or a little more. So there was quite a big overlap. So you could get away with doing like 2 by 150 for this region and still having, you know, if you trim a little bit off, still get like a 50 base pair overlap. That would still work. This particular data set, I had a large overlap. All right, all good? Ready to move forward? Okay, no one's saying anything, so I'm gonna take that as a yes. So the next step we're gonna do is build a phylogenetic tree. And again, we need to do this in order to do the diversity analyses. So we can perform a multiple sequence alignment with the MAFT program. So comparing all the sequences together. And for time's sake, I'm going to just copy and paste this. So we're just importing or inputting our representative sequences and then outputting this aligned file. And then we'll mask the alignment. So what that does is it kind of masks the regions that aren't really informative for determining that phylogenetic tree. All right. And then there's 2 more steps we're gonna do. We're gonna take that filtered alignment and we're going to build an unrooted tree in fast tree, and then we're gonna midpoint root that tree. Yep.
What does that mean, midpoint root that tree?
So how would you, it's basically choosing like a point and then all of the distances are in relation to that one point instead of just to each other. All right, and then rooting that tree. Is it still going for you guys?
Yeah.
It's still going, okay.
masking the alignment?
So there's parts of the sequence that are going to be like hypervariable and will give you a lot of information about the phylogeny, and there's certain parts that aren't really that informative. So
we're just using that portion of our tree.
Yeah.
So this is just like eliminate conserved areas.
Mm.
That's what it does. Okay.
it just makes it faster because you're
There's no, it doesn't have to analyze it.
Yeah. Is this making sense so far? Are you following?
Yes.
Okay. Okay, most people are looking up. Does that mean we've got it up to this point?
Oh, no, I'm still talking.
Okay. So while that's running, I'll just explain the next step. So QIIME has a bunch of diversity analyses that are built into this core metrics phylogenetic method. So there's a couple different alpha and beta diversity measurements that it does. So it does Shannon diversity, observed OTUs is what it says, but again, we're not working with OTUs. They're just, so they're ASVs at this point. Base phylogenetic diversity improves evenness, and then for the beta diversity, it gives Jacquard distance, Bray-Curtis distance, and then weighted and unweighted unifrac. So based upon the question that you're asking, you might want to use different diversity metrics in order to characterize your data. So, but there's tons of them available in QIIME. For an exhaustive list, you can go to this, this link right here, and I'll tell you all the different measurements you can do. But we're just going to work with the default ones for now. How are people doing now? Are we ready? Yeah, okay. So the next thing we're going to do, so we're going to run the default core diversity metrics. So QIIME diversity, and then this core metrics phylogenetic method. We're going to input phylogeny. We're going to input that rooted tree that we just created. We are going to input, we're going to input the table that we got out of the data 2 step. And then there's this P sampling depth. So when we were looking at that denoising stats file, and it told us how many sequences were left after that denoising step. So the lowest one had about 25,000 sequences. So I'm going to sample just up to 25,000 sequences. So I'm comparing the same number of sequences for every sample. We're going to give it the metadata file. So you're going to see we're going to be able to characterize our data based upon different metadata columns. And then give it an output directory. So this core metrics results folder. And if you're typing it out, you might make some typos. Okay, And when it works, it should give you, should say, all right, we made all of these different, so some of these are QZA files, some of them are QZV files, and these are all just different metrics I run. Did it work for people?
Can you go back to like, or can you show more of your terminal window of like what you typed? And then like previous, before these like saved features. Just like, yeah, like here.
So I made some typos, but. Is anyone stuck? So let's say you have, 20 samples, and one of them, just the quality was really bad for some reason, and you, everything you have 20,000 sequences for, and then that one sample you only have 5,000 sequences for, and you want to just exclude it from your analysis, then you can put your sampling depth, say, at 20,000, and then all of these diversity metrics will be for all the samples except for that one that had a lower amount. Okay, so let's move on for those of you who are with me here. So we're gonna look at the alpha diversity metrics. So we're just gonna do one just as an example. So looking at the faith phylogenetic diversity. So what we're gonna do is we're gonna input this faith PD vector.QZA, and we're gonna turn it into this visualization. Because this is a lot to type out, I'm going to copy and paste it. And then once we get that QZV file, we can use QIIME Tools View to view that. And so the output that you're going to get from that are these box plots for faith phylogenetic diversity. And so we can see, so we have 2 samples for microbial mat, 2 sediment samples, and 2 water samples, and that the diversity changes based on the environment. But up here, we can change the column of metadata that we're looking at. Say we want to look at time series. So all these samples, one was early, and then another sample was taken a couple months later. So we can see how the diversity changes over different metadata values. Okay. And then, so you can do that for evenness, looking at a different, different diversity metric, or we can now look at the beta diversity. So we saw that there were a couple QZV plots that were generated by this method. So those we can view in Emperor. So Emperor is like this graphing program that they have implemented in QIIME. So let's look at, so let's do QIIME Tools View and look at the unweighted unifrac Emperor plot. So go into that core metrics results folder, and we're going to look at unweighted unifrac emperor.qzv. So it's going to pull up this Emperor program, and then you get this PCA plot of our samples in 3-dimensional space. And then you can color it by your metadata file again. So let's do by environment. And we can see that our microbial mat samples are most similar to each other. Water samples are most similar to each other. And then sediment samples are not as closely related, but they're more similar to each other than everything else. So that all makes sense. So that's great. Was everybody able to view the alpha and beta diversity plots? Yep. So you would create these QZV files and then transfer the QZV files, so SCP or something, onto your local drive, and then you could use the website to drag and drop it in. Yeah. All right, so I mentioned before, we can see if we've captured most of the diversity in our samples by looking at rarefaction plots. So that's what we're going to do now. So we're going to input the rarefied table. We have a table that was output from data 2, and then we rarefied it when we ran the core metrics analysis. And then this P steps, that's how many steps in that, how many times it's going to subsample when you're making your rarefaction plot. So the default is 10. I'm just doing 25, so we can really see how we can look at it in more detail. But the more steps you do, the longer it takes. And then once that runs, we can use QIIME tools again, QIIME tools view, and view what those plots look like. So we have our observed OTUs. They're actually not OTUs, but okay. But we can see that. So we were talking about sampling depth before. How many sequences per sample do you really need? And we can see that we very quickly kind of reach that leveling off point. So around maybe 6,000 samples or so, we start leveling off. So probably didn't need 25,000 sequences per sample, but I didn't subsample before creating this tutorial. So there we go. All right, moving on. So I mentioned before, there's a step that we have to train our classifier. So we're taking our database and we're saying, cut it down to only look at this section of the database. And then we're going to use that trained classifier in order to assign our taxonomies. But just note that, so say you're using the UNITE database for the ITS region, it's been shown that training that database doesn't necessarily improve classification, but for 16S it does. So the default is the green genes database, but that's not really curated as much anymore or at all. So I'm using the Silva 132 database. The Silva database also has QIIME compatible formats. So just like with any data that we put into QIIME2, we have to import this data. And actually, we probably don't have to do this step. but just know, yeah.
I'm sorry. Continue your thought.
Oh, I was just going to say just the thought that when we're dealing with new data that we're bringing to QIIME2, so we have to import it. But
can you explain what training a classifier means?
Yeah. So yeah, I'll show you on the next slide. So for the sake of time, don't have to actually import it. So the first thing that you do once you've imported that is extract the reference reads. So these next 3 steps took a long time. It probably took about 8 hours to do this. So again, we're not doing it here, but we're taking the sequences, we're telling it what forward and what reverse primers that we used for our database or for our sequences. And so we're based on that forward and reverse primer set, we're looking at that database, matching where those are and trimming it. So we're only going to have the part of the sequence that was between that forward and reverse primer. And then training the classifier. So we take the reference seeks that were output in the previous step, and we create our new classifier. And this is the database that we're going to use to assign taxonomy to our data set. So then you would use this classify sklearn program, input the classifier that we just created. We're going to take that representative sequences file that was output in data 2. So basically, it has one sequence for every ASV. We'll assign taxonomy to that, so it just takes less time, and then it'll match that ASV to all the other ones and assign the same taxonomy. But then we can visualize what that would look like. So we can actually do this part. So we have this taxonomy file should be in this QIIME2 tutorial folder. So QIIME metadata tabulate. We're inputting taxonomy QCA file, and we're going to output a visualization called taxonomy. It'll be a taxonomy.qcv file. And then we can again view what that looks like. And so this is going to look, if you're familiar with Amplicon sequencing, this is going to be like similar to a biome table. So we have our different ASVs and what the taxon is. Taxon assignment. And then, all right, next step would be to look at this data in bar plot form. So we can do QIIME, taxa bar plot, input the table. So I'm going to input the rarefied table. So we're comparing the same number of sequences across samples. We're going to give it that taxonomy file, the QZA file. Give it a metadata file, and then tell it what we want our visualization to be called. So I'm calling it taxa bar plots. And then we can take a look at what it looks like. So the bar plots that you got out of QIIME 1, I think were absolutely horrendous. I think these are a lot nicer. But you can, so at the domain level, so mostly bacteria, some archaea, and then you can choose at what level you want to look at it. So let's look at it at the phylum level. And then I want to sort samples by environment. So I have my 2 microbial mat samples, then my 2 sediment samples, and then my 2 water samples. And so you can play around with how deep of an identification you want to look at, or how you want to sort these samples by. You can play around with the colors too. Pretty. So yeah, did everybody get that all right? Everybody can see those bar plots and play around with them a little bit. All right. So the next thing I just want to talk to you about here is filtering data. You can filter your data based on a number of criteria at this point. So there's frequency filtering. So you can filter out rare ASVs. So if you want to get rid of your singletons, you can do it this way. Say you only want to look at things that occur 5 or more times. This is how you do it. There's contingency filtering. So filtering out ASVs that don't appear in each sample. So say you want to look at the core microbiome across all of your samples. So what appears in every single sample, you could do it this way. Identifier, just specifically trying to which samples you want to look at. Metadata, you can sort it based on, I only want to look at my sediment samples. So do it that way. Or taxonomy, keep or discard certain taxonomic groups. So things like chloroplasts or mitochondria are often amplified when you're looking at 16S. You can discard those types of samples or sequences from your taxonomy, or say you only wanted to look at the proteobacteria, you could positively look for that group. And then just one thing to note, if you are filtering out sequences, you want to rarefy after filtering. So you're not getting rid of half of your sequences in one sample and then still comparing it to other samples. And you can use this QIIME feature table rarefy command in order to do that. And then lastly, guys, we made it to the end, yay. If we wanted to use this data outside of QIIME2, we can export it and then play with it in R or something else. So does anybody have questions? Did it make sense? Is there anything I can explain more?
I just have a question about when you're following up in R. Are there certain packages that are useful for QIIME outputs or?
I don't think there's one specific.
Okay. I just didn't know if
Yeah.
there's a general consensus.
Not to my knowledge.
All depends on what it gets exported as. So like, did it comes out as a QCA file? Is that what it comes out as when you export it?
When you export it, so if you like
CSVs?
export the feature table, it would be exported as a biome file.
Okay.
And then you can convert the biome file into a text file, and then you could read the text file into R.
And then you can use like vegan for like.
Yeah.
Like,
That's a good one.
That's a package that I would use, yeah.
So there's certain packages for Amplicon data or and like environmental data, but not one that's like QIIME specific. Any other questions? Did I completely confuse everybody? And no one's ever gonna do Amplicon sequencing ever again. All right.
Well,
thanks,
let's
Ben.
everybody thank Kristin. She could have said no to this, and you would have all gotten a much more outdated version of how to work with Amplicon data. So it was really awesome that she could put this together. She also did this all within the last month. So all of this other stuff existed with the virtual machine, for years now at this point. And I said to Kristin, hey, that would be really cool if we could get that in the virtual machine. Can you do all the work to make a PowerPoint that will flow through all of this? And then I'll install it in a virtual machine and then you can give the presentation. And she said, yeah, very enthusiastically. So the fact that she's here and did this for us was really awesome. So that's the end of this session. We have one more session for today. It's now just coming up to 4 o'clock. We're trying to aim for that 5 o'clock finish so that everybody's brains can have some time to recuperate tonight. So we're going to take a couple minutes break. Don't try and go too far or take too long. And then we'll come right back and we'll do our last section for today, where we're going to talk about, in the same way that we talked about a little bit about Amplicons and what we need to do to set up for that type of data analysis. We'll talk about metagenomics on a very broad scale. And like I said, we'll do a little run through about how you can take metagenomic data and pull out and reconstruct full-length 16S sequences from that. And if we don't have any more questions at this point in time, we will just end the YouTube live stream and go to the next section. So if everybody, you can go break now.